As pulse production has increased in our region, so has pulse root rot pathogens, including Fusarium, Aphanomyces, and Rhizoctonia. In this video, I will discuss some of the research being performed by the Plant Pathology Program at the MSU Eastern Ag Research Station. My name is Frankie Crutcher, and I'm the lead investigator for the program. Although my group does a lot of research for pulse pathology, today I will focus on only two projects. The first project examines how Fusarium from scabby seed can negatively impact rotations with pulse crops. When I started at the EARC in 2016, I collected Fusarium head blight affected wheat and barley plants from both eastern and south central Montana. We isolated the Fusarium from the grain heads and went about identifying them to species. Fusarium head blight is most frequently caused by Fusarium graminearum, which is very infectious and produces vomitoxin, also called DON. We did find quite a bit of Fusarium graminearum, but we also found Fusarium avinaceum in large numbers. Fusarium avinaceum has been found to cause head blight previously. However, it is usually not a dominant species and does not produce vomitoxin. It is, however, the primary cause of pea and lentil root rot both in Montana and North Dakota. This discovery led to an important question. If a farmer increases the fan speed on their combine to remove scabby seed from their harvest by blowing out in the field, something that has been recommended in other states, and Fusarium avinaceum is in their scabby seed, how might this affect root rots if the farmer plants pea the following year? We also wanted to look at how seed treatments affected root rot in this instance. To best answer our question, we wanted to mimic what a farmer would do as much as possible. So we selected sites previously in wheat under no-till conditions. To replicate scabby seed, we grew 3 of the Fusarium avinaceum isolates that we collected in 2016 on barley. We then calculated the weight of the seed that would be blown out of the combine if there was 5, 10, or 20% scabby seed. 20% may seem like a lot, but the testing lab in Saskatchewan regularly receives samples with this much scabby seed. We had 2 locations, one at the USDA dryland field site in Sidney, Montana, in collaboration with Dr. Brett Allen, and one at the Southern Ag Research Station in collaboration with Dr. Ken Kephart. In the fall, we marked out our field site and spread our scabby seed amounts in the field. In the spring, we went back to the site and planted our treated and untreated pea seed. For this experiment, we looked at 8 treatments, which included Amethanoxam and Cruiser only, Rhizolex, Trilex, Myrtect, Vibrance Max, Obvious, Rancona, and Evergol Energy. Over the course of this season, we took stand counts to measure emergence and damping off. We measured root rot by a scale of 0 to 5, and we recorded final yield and protein. In 2019, we did not see a difference in stand counts at any timing. However, we did measure a difference in root rot severity between different amounts of our scabby seed at our Huntley site. We also measured differences in severity between different seed treatments. We hypothesized that these differences were measurable in Huntley due to the increased amount of residue in the fields that would be ideal for growth of the fungus. We observed Fusarium rotting through the stem at the soil line in Huntley in a way that was not observed in Sidney. The severity ratings here are relatively low, but the plants were collected at about 35 days after emergence, which is very early in the season, and most likely this severity would continue to worsen as the season progressed. This is also shown by the disease incidence measurements, which is the percentage of plants showing root rot symptoms. Similar to the severity ratings, we measured significant differences at the Huntley site for inoculant percentage and had differences between seed treatments for both sites. How this related to final yield numbers was only evaluated at our Sidney field site, as the one in Huntley was destroyed by a storm just before harvest. We measured significant differences for yield between different seed treatments. Adding a seed treatment in some instances increased yield by 500 pounds per acre. We are repeating this experiment in 2020 to confirm our results. As we only have 1 year of data for this study, we cannot draw definite conclusions. However, our work supports previous studies that show seed treatments are effective against Fusarium root rot and can increase yields. Additionally, we can say that spreading scabby seed infested with Fusarium avinaceum in the field can cause root rot in pea the following season under no-till conditions, and it is a practice that we do not recommend. The second root rot project I will be discussing today looks at the effect of variety on Rhizoctonia root rot. From 2017 to 2019, we have performed seed treatment studies looking at their efficacy against Rhizoctonia solani, a common root rot pathogen. We grow Rhizoctonia on barley and we run it with the seed through the planter. We are using an AG group of Rhizoctonia known to infect both grasses and sugar beets. This isolate is also used in our sugar beet experiments and can cause severe root and crown rot as pictured here. Unfortunately, in all 3 years, we could not identify a seed treatment that was effective against this pathogen. For these experiments, we used Richley lentil, Sierra chickpea, and AAC Carver pea. We observed severe stand loss and yield losses for both lentil and chickpea. In chickpea, Rhizoctonia attacked and rotted the seed before germination could occur. For lentil, we saw decreased germination, but also rotting at the soil line that led to post-emergent stamping off and severe stunting. In peas, we have observed decreased height, but in our seed treatment experiments, we measured no loss of yield. I am in no way saying that seed treatments for pulse crops are not needed or are always ineffective. As shown earlier in this video, seed treatments can be very effective against pathogens such as Fusarium. Additionally, environment plays a big role in severity and development of diseases and cannot be discounted in this case. But because seed treatments were ineffective in our experiments, we decided it would be beneficial to look at common varieties to see if any have resistance or tolerance to this pathogen that might be paired with seed treatments more effectively. Varieties selected for the study are shown in this table. For chickpeas, we selected both Kabuli and Desi types, with the thought that a thicker seed coat in Desi chickpeas might provide more protection to the germinating seed. For lentils, we looked at both green and red varieties. For peas, we selected both green and yellow varieties of different sizes. We tried to include varieties grown in Montana, but we also wanted a selection of Desi chickpeas, so we included Golden Dragon and Black Sicilian that are commercially available but not grown in Montana. As this is our first season, we do not have final data, but we did see some differences in germination rates for chickpeas and lentils. As you can see from these pictures, stand losses from Rhizoctonia can be quite severe in chickpea. In lentils, the stand losses initially are not quite as severe, but damping off is still occurring and stand losses will increase as the season progresses. In peas, most varieties do not visually have dramatic stand losses, but some plant death is being observed later in the field. It will be interesting to see how this affects final yields. In summary, it appears there are varietal differences for resistance or tolerance to Rhizoctonia solani that might be beneficial for future breeding efforts. We will continue this research to verify our findings and in the future test whether pairing resistance with seed treatments leads to better fungicide efficacy. I would like to thank the EARC Plant Pathology Program staff and students for all their hard work this year. Both of the projects presented here are funded by a Montana Department of Agriculture Specialty Crop Block Grant.